Thank you very much, Rachel.
Yes, absolutely. I am honored to introduce today's speaker, in which we're very thankful to have him here, Dr. Simon April-Monn. Dr. Simon April-Monn is a postdoctoral scientist at the University Hospital Zurich and the University of Zurich in Switzerland, and he specializes in personalized cancer medicine and precision oncology. His research delves into understanding the regulatory architecture of rare cancers, including neuroendocrine cancers, aiming to develop personalized therapeutic approaches. Committed to open science, the 3R rule, reduce, refine, and replace, and ethical research practices, Dr. April-Monn employs innovative experimental models and advanced in vitro systems to decipher the complex biology of rare cancers. Through rapid ex vivo drug profiling and patient-derived cancer organoids and computational techniques, he seeks to identify individual drug sensitivities and translate his findings into clinical applications to benefit cancer patients. So today, Dr. April-Monn will be talking to us. His talk is titled From Cancers and Disease Models, Insight into Patient-Derived Cancer. So thank you, Simon, so much for coming, and I will pass it over to you.
Well, thanks, Rachel. Thanks a lot for the kind introduction, and also many thanks to the entire NETChatter Foundation board and the NET community to have me here. It's a great honor, and I'm looking forward to reach out in this NETChatter format. As you mentioned, well, I'm particularly interested in personalized cancer medicine and precision oncology using ex vivo cancer models, because I truly believe that such disease models can help us to add a functional perspective and help us to find effective anti-cancer therapies, and hopefully will help us to find cures for cancer patients. And that's also the reason why I actually will approach this talk not purely from a neuroendocrine perspective, but rather from a pan-cancer disease modeling perspective. And it's actually quite a nice coincidence that, and it's also a great honor, but also a bit of a tricky task to follow up after Jacobo Michael's fantastic talk last month on neuroendocrine cancer disease models. I don't know if I can keep up, but I will try to do my best to share my view here. So let's see how the program looks like. So I want actually to break down the topic of today's talk into a small and simple concepts that should actually also address and invite a more general audience. So and obviously I'm happy to discuss more details and in the Q&A, and if you have some questions, you can drop me an email anytime. So in the first part, I will talk a bit about disease modeling, some general aspects in regard to cancer research. In the second part, I then try to give you kind of a virtual lab visit to show you what is literally under the hood, and this is work that I've been doing mainly during my research at the University of Bern, and it has been a great collaborative effort with many great clinicians, with scientists and many other supporters, for which I'm very grateful. The third part then is more about how we then applied that model in a proof of concept study. And in the fourth part, I want to open up a bit and looking beyond the box and beyond the ivory tower, and I want to very briefly summarize where are we in the NEN field. I want to show you 3 brief examples of success stories from other cancer fields, and I want then to wrap up with what's being done in the NEN community. So that's the program, and I would say we dive straight in. So I want to start off with a quote from Professor Box. So he was a former lecturer at the University College of London, and he's a great statistical mind of the 20th century. And this quote has been used many times, maybe even a bit inflationary, but I really do think in the context of that talk today, it really makes a lot of sense to bring it up. It says all models are wrong, but some of them are useful. And I think on the first read, this might sound a bit discouraging, because we want to use models to understand what's happening around us. So if all of them are wrong, well, that's not the best ground to start with. Now, what we are trying to achieve via modeling is actually to break down the complex real world into a simpler, understandable format. And that actually allows us, as I said, to make sense of what's around us. And I think everyone of us is using models every day, and we heavily rely on the output of many models in our daily routines. So, but if all models are wrong, well, why do we actually bother modeling then, right? And I think this comes down to understanding if a model actually is capable of capturing all the relevant information to answer the question that you have. So, for example, if you want to do a hike in Switzerland, what you're actually doing is you usually use a map, as I did just a couple of weeks ago, and use a map to navigate through the terrain. Now, if you want to know more about the weather, you might use, you might not use the map, but you rather might use the Swiss Meteo app to understand what's the weather I'm looking like. And in essence, what we are doing in disease modeling is an analogous situation. So we're trying to break down complex disease, in this case here, cancer, and we want to break it down into simpler and controllable and testable formats. The most common models that we typically use in the lab is, for example, immortalized cancer cell lines, which we usually refer to in vitro modeling. We use genetically manipulated mouse models, or GEMs, which is referred to in vivo modeling. And more recently, there has been a trend towards patient-derived tumorids, or PDTs, or cancer organoids, depending a bit on what setup you use, which I would refer to ex vivo modeling. So obviously this is a simplified version, and there is a lot of great reviews on the pros and cons of all these models, and I can encourage you to check out Dave Duvison and Hans Kleber's review, or Muni Zhang's and Jesse Boehm's review from 2019. Which give you a lot of details. What's the difference between these models, and why they are good in certain situations and why not in others. But I want to highlight that again that these models, they are not the actual cancer. So we know that mice are not men, nor are cancer cell lines or patient-derived tumorids cancers. So these are all models and approximations, and it's one of the most difficult tasks as a scientist to find a good and a relevant model, for all the questions that we still have about a disease. I think if you have wrong or irrelevant models, this is very difficult to answer your questions that you have. So with that background, if we jump straight into the topic of neuroendocrine cancer, one of the major challenges is that we are missing representative human NEN models for preclinical research. And I think this hinders the translational advances. It hinders functional and mechanistic insights, which are needed also in the clinics to treat patients. So what we literally have is kind of a handful of cell lines, such as BON1, QGP1, CM, and NT3. And I think in looking at that picture, it's even it underlines how important Daniel Benton's and Jörg Schrader's recent work was on the NT3 and other NT models, because we really do have very little cell lines that we can use in the field. Now, if we compare that to another, cancer entity such as breast cancer, you can see that the quantitative lack of model is quite drastic. I think it's, but not only about the quantitative aspect, it's also about the qualitative aspect. So when you look at the very or a mouse model that is often used in the field, it's called RIPTAG2 mouse model, which actually has the red insulin promoter tagged to an SV30 early region tag. which then actually inactivates RB and P53 in the mice and leads to cancer. So if you look at that model, we know that mice are not men. And actually, in 2015, Angurar Sadanandam and Doug Hanahan from EPFL actually have shown in one of their papers that in this mouse model that is often used, we do not find all the human subtypes that we face with patients. So as you can see in this heat map here, that blue category, the human intermediate MEN1-like subtype is not represented at all. And it's a similar picture if we look at the available cell lines. So again, here, this intermediate MEN1-like subtype is missing. So without a doubt, these models have been very, very helpful and have advanced our field. However, it's still a bit on debate, what subtypes these models represent and to which category they belong. And it's also not clear whether these cell lines and the mouse model actually represents the disease heterogeneity or the patient situation. So I tried to tackle this challenge from a different perspective, and I tried to find a way to model the patient situation from a slightly different angle. And for that, we actually built an N network where we collected patient material and fresh frozen donor tissue. And we then actually processed that material using next generation sequencing, using next generation TMAs. We did ex vivo drug screenings, and we did we also collected clinical and the follow-up data. And we actually wanted to see if we find some association between these layers here. And I will now guide you through the modeling part and the virtual lab visit, and I will then follow up and just briefly show a preclinical application that we did using that setup. So let's have a look at the lab and to start with, as I said, well, it's difficult, or NEN, I think we know it is a rare disease and it's difficult to collect sufficient material. But it's also historically the case that it was challenging to culture these models in classical 2D monolayers. And what was interesting is when I started my project, it was interesting to see that actually already in the 80s, there were some reports that the pancreas and especially the islet cells, they have kind of a specific architecture. So rather than sticking to the plate or sticking to a 2D surface, what they would do is they form these three-dimensional aggregates, and that was interesting to see because that gave us a hint that maybe such a setup would help us to come up with a model. And if you look at that in a picture way, you can see that there is a drastic difference. So here you see MCF7, that's a cancer cell line. And if you compare the 2D setup here on top with the 3D, you can actually see that it's a big difference whether you use setup 1 or setup 2. And I don't want to go into too much details here, but we do not have any pre-described polarity in the 3D culture. And what is also important is that we have so-called soluble gradients if we culture the cells in 3D. And that can be, oxygen gradients. It can be gradients of nutrients or metabolites that we find in these spheroids. We also have zonal patterning usually. So at the outer ring, you have proliferating cells. You have more quiescent cells in the middle, and really in the core, you usually have a so-called necrotic core, so where the cells are actually dying because they don't get sufficient oxygen or nutrients or metabolites. So how do we actually culture patient-derived biomaterial in 3D in the lab? What we actually do is we collect the tumor donor tissues, and we then digest them into a single cell suspension, and we put these into so-called free-floating suspension culture using ultra-low attachment plates. And what we also do is we then add growth factors which are specific for GAPNENs. And we also add a low percentage of extracellular matrix to actually allow the cells to come together and form these spheroids. And that's how it looks like, not in a schematic, but in real time. So you have these ultra low attachment plate, you have these U bottom plates, and if you look into them, you can see these small clusters, basically the cells coming together. And if you then again zoom in, you see these spheroids look more or less like this in culture. Now, when we look how this looks in real time, let's see if that works. I will just use that one here. Let me just check. I'll just switch. Where is the pen? Let's see if it works. If I just click, no, it doesn't work. Ah. Let's see if I can get that to work. I'm checking if I can get the pointer, but it doesn't seem like I have. Ah, here, pointer, pen, highlighter, ink color, arrow options. Ah, now, patience. You just need patience. Okay. So you will see how these cells, aggregate together in that in these plates over a longer time point. And I think it's important also to imagine how this looks like. So it's approximately 200 something-ish hours. You can see that in time lapse now. Okay, let's switch back to the laser pointer and never touch a video again. So, okay, now, so you see that these cells in the time lapse here, it's a bit easier. You can see that after 72 hours approximately, you have these three-dimensional structure formed, and they stay relatively stable over a long time in culture. Now, what you can also do is you can take them out and you can stain, basically cut them and stain them, and what you will see.
Simon, I'm so sorry to interrupt. Are you sharing your screen? I'm not seeing it, or maybe just on my
sorry. Yeah. So,
yeah.
no, I'm not sharing anymore.
Sorry about that. I was.
So did you see? But you saw the video, or
Yeah,
not?
I can see it.
You see it?
Same
Yeah,
here.
we can see it.
Oh,
Okay.
that's maybe just me, or okay.
I think I share, but I share again. So
I see
Okay.
slide 13 right now.
Okay.
yeah. That's the slide.
Okay.
So all because of this video. Never put a video in.
The video was
no.
really cool.
Okay. So yeah. So they come together and they form these aggregates. And I hope, Heidi, that you can see it now again.
Thank you.
Okay. Good. So yeah. So if you look how this, yeah. So you can take them out, and what you can do is you can basically cut them and stain them. And what we do usually is using H&E stainings. And you see here one patient. And just to bring you on the same page, what we are talking about, the size. So this structure here is basically 0.5 millimeter approximately, which is half of the size of a very sharp pencil. So that's the size where we're talking about. Obviously, we magnified it, and when you look at these structures, you can see it's not just an aggregation. I think it's a symbiosis to some extent. So it's a complex spheroid structure, and you can see that some cells for these tubular structures here. You have some potentially some ECM excreted, and you have some rims outside, which is actually showing that it's a relatively complex symbiosis which is happening in these cultures. You can also stain those cultures using, in this case, synaptophysin, so a clinical biomarker for neuroendocrine cancers. And then you can see that you have mostly neuroendocrine cancers in these cultures, but you also have some other cells which we think is kind of the secret, maybe that this actually works. So You can also then look at different patients. So again, this is patient 1, which you just looked at. But if you look at, for example, another patient, you can see that these structures look a bit different. And if you look at another patient, you can see that the structure look again very different. And I think this actually reflects the patient heterogeneity, and it might reflect even the patient situation in situ. Well, now we have all these, fascinating tumorate structures ex vivo, and that can be used and applied to model neuroendocrine cancer. Now you might ask, well, now what can possibly go wrong, right? So you have the models, and no problem. But in fact, I mean, it took, and it still takes us a while to figure out how we can consistently get these structures. And we came across surprises, and I think the group still comes about certain surprises on a daily basis. So what can possibly go wrong? I think one of the crucial aspects that I learned from doing these cultures is that actually the quality, I mean, as simple as it sounds, it's actually the quality of the biomaterial and the donor tissue which we receive in the lab, which makes a huge difference. And as you might imagine, this depends on many factors and many players throughout the workflow. So what we try to do is, with the time when we knew what's more or less happening to these cells, we built in many quality control steps, which I think are very important for any drug screening. These control steps, such as these microcell blocks, actually allowed us then to detect cases and potentially exclude cases from our analysis. And in this case here, you can see you have a patient which came with a lot of necrotic cells, so the necrotic core. And if you isolate this patient, you don't have to be surprised that you have very low viability in the cells. And then in this case here, we had a patient which had some extracellular matrix and built these structures here, and that was actually also reflected in the culture. So you see that all these blue dots are not stained for synaptophysin, so indicating that these are other cells. And obviously we don't want to just screen other cells. So these are cases which you have to analyze and then take out and show the people that sometimes it can go wrong, and that's the case here. Then if you zoom out, there are also other factors which I try to consider, we have scarcity of biomaterial in the donor tissue. So usually what we get is roughly 1 million cells per sample. So you have to really streamline the setup and prioritize the treatments, and also prioritize what questions can you answer with your model. And then I'm talking about success rate. So we came across, what is a culture success? How do you define that? And we also had to show that to the reviewers, what is actually our understanding of a successful culture. And this can differ very drastically. So we try to be as transparent as possible to say, well, what is actually a success? And then you have some other factors like the media composition, the preparation of growth factor stocks, and the stability of growth factors in the media and in the culture condition of 37 Celsius. And you have consumables and supplies such as matrigel and the plates, and during COVID and the supply can break down, and then you have a problem. So that's something you just have to keep in mind and think about when you do such assays. I now want to come back to the topic of personalized medicine, and I think it's an upcoming field and has many perspectives and potentially also raises a lot of expectation. And what I mean by personalized cancer medicine is actually that you would actually treat every patient with the right treatment at the right time. So now this definition can vary, and within the field you might have different flavors of that idea. But I really go for that aspect. So, and I think it's very helpful, especially for rare cancers, for difficult to treat cancers, and for cancers that are heterogeneous, such as neuroendocrine cancers. And what we do is, we form these three-dimensional structures or spheroids, and then you would actually perform drug screening and genotyping in the lab. And ideally, you then have you can provide some supplementary information to the clinicians which might help them, to decide which treatment to go and which not. So that's what we actually did in the proof of concept study. So we went to high-grade GAP9 patients, and we collected samples where we had the therapy and the follow-up information. And we then also wanted to have cases where we have the tissue and the cryomatched biomaterial. And I have to say it's a very rare case, because usually these patients are in palliative care, so they don't get a lot of sample removed. So it's a rare case. And what we actually did is we looked in different tissue biobanks, and what we found was basically 8 cases where we had all this information complete. What we then did is we looked at the genomics, we looked at the transcriptomics, and we did some drug screening with the model that we had. And this has been done in many other cancer entities as well, and we were not the first one. But I think what was really our strength was that we combined our extensive clinical knowledge with the functional drug screening, and this was at that time a relatively new combination. So in the study, if we look at the samples again, you can see the original tumor rates. Again, it looks very similar on the morphological aspects. But also, if you look at the transcriptional level, what you can see is that the original tumor rates and the PD tumor rates, they're actually clustering together in this dimensionality reduced space here. And what is interesting is if you compare that to the non-cancer cell lines, you can see that they're actually clustering apart, which indicates that, at least on the transcriptional level, the non-cancer cell lines are not very representative of the tumor rates or the original tumors. We then performed that drug screening, as I mentioned, and we looked at cisplatin sensitivity, and we divided the cohort into low and high sensitive patients based on GR metrics. And what is interesting, if you look at the ex vivo drug response that we measured and compare that to what actually was happening in the patients, you can see that where the patients got the treatment, you can see that this is actually a perfect match. Which indicates that the model at least points towards the right direction, and obviously the numbers can be higher, but for our cases it looked very promising. We then also did some more modeling. I won't go into detail now. You can read that in the publication. So it was circling around the cisplatin stress response on the transcriptional level. We then did some target interference in silico, and we also did a rapid ex vivo drug screening, looking at drug combinations from the targets we derived here. That was actually our study, and I think there are some quite intriguing insights that we were able to bring out of that model that we have. So now in the last part, I want to open up and give some perspectives. And coming back to the topic again of personalized cancer medicine in NENS, well, where are we now? And I think it's fair to say that in recent years there has been a tremendous amount of work put into these aspects, and many great studies have been published and point towards feasibility of that personalized cancer medicine approach. But as with many things, there is still a long way to go, and to translate all these findings into clinics, and some of them might not be translated, some of them might, we don't know yet. And well, you might wonder, well, why are you not quite there? And I just scribbled down some points to consider, and I think one point, zooming out of our study, is really the interaction with the ethical committees, and it's very important that you protect the interest and the rights of the patient, but this takes time, and you have to discuss, and you have to look at the studies. Also, very important, the patient consent, so that you inform the patients about the donor tissue usage, that you inform them about the signed general consent, and how important it is for us as scientists to get the material, to actually look at it. That's another point which also the hospitals, at least in Switzerland, are trying to synchronize and update. Then another part is just the logistics. So between research institutions, it's about material transfer agreement, between universities worldwide. It's sometimes not very easy, because you're basically shipping, biological material. So and that's something which you cannot just send via post. So it has some things, some administrational tasks that you have to do. And then, you just have to streamline the biomaterial processing. So that means that it's important to establish and also to access tissue biobanks. It's the communication between the clinicians, the staff of biobanks and the laboratories, which is also very important. And, to set that up and bring everybody on the same page, this takes time. And I think these are important points one has to carefully consider. And yeah, this takes time. That's why we are not there yet, as one would like to be. So still, what I want to do is now to switch gears to other studies and other fields, and I want to just briefly touch on 3 successful co-clinical trials and using patient-derived human models. And one of these studies is the tumor profiler study at the University of Zurich and the ETH-SET, where Anja and Mitch and Gunnar actually try to do a co-clinical trial, and they show very interesting stuff and data where you can see what you have to consider if you want to do this in parallel. And there is a lot of hurdles that you have to take, and I think they did a fantastic job in that. Now, if you go a bit up from Zurich north to Basel, there is this, there is Roche, the Institute of Human Biology, and they are really pushing forward and advancing the human model systems, and that's also an interesting approach. And they're highly motivated and have very talented staff that actually pushes that field as well, which I think is a high success story for the patient-derived human models. And then if you go again a bit north, going to the Netherlands, you have the Hub Organoid platform, which also provides some services of patient-derived models and 3D models. And I think all in all, this shows that the personalized cancer medicine or the precision oncology approaches are actually taking off, and also are translated into applications. And then what's being done in the NEN community. And I think although the neuroendocrine cancer field is not the most common cancer, the community is extremely active, and I find this very fascinating. And what came up was this NET model consortium. So that was Dr. Kate Lyons and the founding group, which actually looked at the nomenclature, the taxonomy of NEN models, the resources, sharing resources, material transfer agreements, and so on and so forth. We have the NETRF. It's not just fundraising for research projects, but it's also providing a lot of information and educational resources for patients, families, and caregivers. And then, you have a lot of other patient advocacy groups, such as the NET Cancer Foundation. And I learned many more groups are out there. So John Metzger was a very interesting insights that he showed me, just the tip of what is actually going on. And I was not aware as a scientist, but I think it's very important to keep the field rolling and advance the knowledge. And what I did is I just put together a list of links. So for people who are interested, you can check them out and see what is actually done, from providing newsletters to support in the educational nature direction. So webinars, literature, there is peer support, there is, in-person, online meetings, support groups, and also political advocacy, which I think is very important also for the community. And so I want to finish up with the notion that, relevant models are useful. And I really do hope that our work belongs to that category and will help to advance the personalized cancer medicine and precision oncology field, and to help patient and find novel cures. And obviously there were many people involved, and so my former group and the entire endocrine pathology group in Bern. And I also want to especially thank our collaborators, without whom it would not have been possible. So kudos to Massimo Falconi and his group in San Raffaele, also to Bertram Wiedemann and Katharina Detjen at the Charité Berlin, our surgeons, Beat Glor and Corinna Kim-Fuchs, and oncologist Attila Kolar at the University Hospital. And I want to thank you for your attention, and I'm happy to take questions and discuss more about NEN disease models.
Thank you so much, Simon. That was an excellent presentation. So at this time, if anyone has any questions, feel free to put them in the chat or go ahead and unmute and ask. I have a quick question, Simon. Is the video published? I'd love to show my students one day. Have you published that video? Is it in the paper?
Yes. Yeah.
That was on the slide.
Yes, that's in the neuroendocrinology publication. So it's a bit hidden in the supplementary, but you find it. I think it's
wonderful.
On Figure Share or something. Yeah.
Thank you.
I'll ask a question as well if no one
Yeah,
else.
sure. Hi, Kate.
Hi. So one thing that I've had discussions with people a lot, and I was interested in your opinion, is do you think we'll ever get to a state where we will be able to get cells from a patient and test these things out and then be able to sort of directly feed that back into the clinic?
Well, I hope, and I think this has been done, until now, not extremely successful, but it has been done. I think a lot of groups from the Netherlands are really pushing this direction. I don't know about the neuroendocrine field. If I hope, I really do hope, and to do that, you just have to be aware that. What approach you're taking. So to feedback it directly to the clinician, it has to be relatively in a short time. So I think that would opt. So I would opt more for rapid models for that aspect. But if you want to understand more about the disease and the mechanism, you might go to the long-term models, such as cancer organoids, which probably would not feedback directly to the clinicians, I think. But yeah.
Great, thanks.
You're welcome.
So really Simon, oh, sorry, go ahead. Was that Stephen?
Yeah, it was.
Yeah, go ahead.
Really, no, sorry, Rachel. Really good talk, Simon. It's really interesting model base. Did I catch that correctly? You do most of these things fresh, so kind of from fresh samples, or are they from mostly frozen? Like kind of patient samples that have been cells that are frozen, then you kind of bring them back. So which one do you kind of lean more towards?
I think this was so we set up the pipeline that we can use both fresh frozen and fresh material. But for us there was no difference between using fresh frozen and fresh material. I mean, you get a bit more yield when you have fresh material, but it works quite nicely with the fresh frozen material. And I think that's an advantage to know that it's possible, because then you can ship and you can collect samples. You don't have to, stay up for, I don't know, like late or whatever. So you can actually collect them and screen a collection of samples, which I think is quite handy and helpful.
Yeah, I was asking because I could see this really, the tumoroid kind of rapid testing model really being useful in the frozen kind of field, because just from being able to get more patient-specific information, it help guide your drug screens downstream. So the big issue with doing these fresh, obviously, is you don't entirely know which things to test right off the bat unless you really get good sampling kind of quality from the patients early on. Maybe they have a preliminary genetic screen. So I could see a lot of utility in keeping these frozen and then testing some of that tumor against just to know either genotyping or looking for functional kind of activity, and then being able to later guide that treatment downstream. So that's really where I kind of see a lot of advantages for the tumoroids. Yeah.
And you could even add that up. We use a simple freezing system, but if you take a look at the embryology field, what they do is this vitrification. So you take the eggs, the eggs of the females, and you can actually preserve them. So you could also go into that direction and even preserve that better. I think what is important is that you streamline the exchange with the clinicians, so with the surgeons and with the pathologists, that the material is not just laying around the whole day or over the weekend. But if you get that, I think it's not even necessary that you have to do very fancy techniques to preserve the material. But that would be a direction. If you want to have the best quality possibly, you can also maybe use that one. Yeah. But
yeah, thank you.
Giovanni, I see you have a raised hand.
Yes. Nice presentation. I have only one question. Have you found any correlation between the Ki-67 in your tumoroids with the histology of patients?
Yes, we do. So actually, what we did is we correlated the metabolic surrogate that we had from RTG readouts with Ki-67 and the grading, and we see basically a perfect correlation, which is quite nice. So you know, what we see in culture is associated what the patient, you know, has in terms of Ki-67 indices.
Okay.
Yeah.
And another question. I've seen that there is something about interferon beta in one slide, but
Yes.
it was very,
Yeah.
very fast, so I wasn't able to read. Do you have any experiment on that also in patients?
So what we did is we used interferon beta 1, or it came up as a top target so we tried to infer potential combination targets, and interferon beta and KDM5A came up as top targets. So what we did is we actually combined interferon beta, which was, you know, I'm not a clinician, I'm not an oncologist, but it has been used previously as a compound to treat neuroendocrine cancers, and we used that in combination with cisplatin, and we found that it actually had an effect. It was not just potent, but it was synergetic with cisplatin. So we did that ex vivo, and that's, you know, basically the last part of our findings that I went a bit fast, but yeah. Okay. I think it's an interesting aspect because it could be a potential treatment for, you know, NETG3s, where you don't have a lot of, it's not so clear. I mean, but yeah, you have to ask the oncologist, but okay. Maybe I'm just a naive scientist, but.
Thank you so much.
Samira, did you have a question?
Yeah, but I know we don't have much time, and you had questions too. Just very quickly, excellent talk, Simon.
Thanks, Samira.
Look forward to all your future work.
Yes.
Quick question. So, you know, you're talking about the translatability of this. I guess from a clinical standpoint, right? Do you think at large that the grade ones or lower grade ones that don't grow, at least we have a hard time having anything to grow. You basically, I guess in my approach, you could, I guess treatment would be surgery, so you don't have to do many drug studies for them. So in a way, your model is perfect. If you looked at the grade 3s, it's where we need additional therapy. And I'm sure many oncologists have more input on that than myself. My question to you, you know, we're working obviously on organoids, but also on a patient-derived model. I don't know if you have experience with that. In that one, I don't think it's going to be very quickly translatable because we basically wait almost 6 months till we see anything grow. Any experience with that in your hands?
Yes, obviously it's easier if you have high graders. Obviously that makes the whole setup a bit more you know, easily, or maybe a bit more easy translatable. But we also screened the low graders, and what is difficult there is that you know what is a good readout. So you know, if nothing is happening, if cells stay viable, that's a good sign. But you don't know if if they're just sitting there, or you know. So we try to screen, and obviously with those samples you cannot screen, you know, compounds that need a lot of cell division, such as cisplatin or you know, some other alkylating agents. So I mean, it would be interesting maybe to test some SSTR or somatostatin analogas, but that's not what we did. But I think that's something which could definitely be done. It's just you have to be aware that, you know, the cell sitting there being viable is good, but you don't have a lot of changes. So it's difficult then you can just see a decrease maybe in viability or in cell counts, but you don't know if the cells, you know, what they are doing. So yeah, definitely more difficult. And I would say, you know, you can probably cross out the G1s, and maybe it's better for G2, G3. Yeah.
Great, thanks.
Oh.
Hi, great,
Hi, Paul.
fantastic talk. Hello. Yeah, that was amazing, Simon. Thank you for sharing and thank you for being here. I just want to follow up on it, and you're definitely like like the pioneer in this field, like taking us through all these almost impossible tasks that we weren't able to do. And I just want to touch upon the it's always challenging to get them to grow in the longer term, and despite our success with like these short-term culture, have you do you guys have any improvement to help like get them past like the 3-month stage and all that to get them grow a little bit better? Because from what we see as well, like it's like after the first month and the third month they're hanging in there, and then afterward they just kind of like go into a phase that they don't grow anymore.
No, I mean that was not our questions, or what we had some high graders which we just kept because they were growing a bit faster, but it was not the aim of our study. So we tried to really come from this short or rapid ex vivo drug screening, and in that regard, it's obviously Talia Dayton who has done a lot of work on their model. But I think as I understood, she had a hard time also, getting them to grow. What we have, the compounds EGF, FGF, placenta growth factor, and PDGF is, this composition maybe helps, but I don't know if it's the best medium to keep them for long term. So we did not really go down this rabbit hole. We tried to keep it short, and yeah. So, but there is also the approach from Muni Zhang, which tries to have a 2D phase and then you go 3D again. So I don't know. I mean, would be interesting to test, but I think it's hard. So if I understood Talia correctly, it's a hard task. So.
Yeah, well, thank you. Thanks for sharing the insight.
You're welcome.
I think my question was actually answered in this discussion. Did anyone else have questions before we wrap it up? Yeah, it's definitely a difficult task to get these cells to stay alive and then, yeah, of course, divide. Because in my experience, I actually had them alive for 9 months and they just never divided. And it makes, I mean, I had to stop checking the viability because you take some cells out to check the viability, but then they don't replenish. So it was like a dwindling population. And I'm always think to myself, like, what is the missing ingredient? And I don't know if a missing ingredient is the answer. Especially, Simon, it was really interesting how you talked about kind of the architecture and the shape and how, we don't have that polarity in 3D and how that's affecting in 2D. And the islet cells and the neuroendocrine tumors, their affinity to grow in like a nest, maybe there's some structural, it's not necessarily a missing ingredient. There's also a structural aspect to growing these tumors.
Yeah, definitely might be. Yeah.
Well, all right. I think we can wrap it up if there's no more.
Sorry, Rachel, can I just butt in for 1 second?
Of course you can.
It's not a question, but what I was going to say is I noticed there are a few people on this group that maybe haven't been involved before. And so the Net Models Consortium, Simon sort of touched on, and Rachel is actually helping us out with that anyway and is kind of one of the co-founders and helping. But I just wanted to say that if anyone is interested in getting involved in that, it's an open club, the more the merrier. So I will put my email address in the chat because I've recently moved jobs. So not necessarily everyone knows my email address. And if anyone is interested, then please drop me an email and you can join the sort of mailing list. And we're kind of having an annual meeting as well where all of this stuff is being discussed in hopefully great detail to help us move forward. So I will do that.
Can I just say, make sure you save the chat, because otherwise it will disappear. Once I end the meeting, the chat will disappear. So you need to press save the chat button. So Kate, if you put your email address quickly in the chat now, and then people can save it, and then they've got it. But while Kate's doing that, just like to say thank you very much, Simon, for putting together this talk. It's been really, really interesting, even to me that knows nothing. So I'm really, really grateful. I'm grateful to everyone who's turned up to listen. So thank you very much. And again, remember to save the chat with Kate's email address. And our next meeting is on the 27th of June. We're back to the last Thursday of the month again. So my fault we were early this month because next week's my birthday. So, so thank you very much, everyone. Thank you. And I will see you all next month. Thank you again, Simon. Thank you.
Thank you, Selena. Thanks a lot.
Bye-bye.
Bye-bye. Bye-bye. Ciao, ciao. Bye-bye.
Bye-bye.
Bye-bye.